All right, let's dive into one of the most fundamental, most elegant processes in all of biology. It's this intricate, absolutely essential dance that allows life to even exist. We're talking about the cell cycle.
Okay, so what you're looking at here is basically the blueprint for a cell's life. Now, it might look like a simple little loop, but this cycle, this is a masterpiece of biological engineering. It's responsible for growth, for healing, for the continuity of life itself. And, it brings up a pretty massive question.
I mean, really think about it. How do we go from one single cell, a fertilized egg, to the complex, thinking, breathing beings we are today made of trillions of cells? Well, the answer is right there. In mastering that process we just saw, over and over and over again with incredible precision.
At its heart, the cell cycle is the engine that drives all complex life. I mean, right now, as you're watching this, millions of cells in your body are dividing. They're replacing old skin cells, they're healing that cut you got yesterday, they're keeping your organs running. It's this constant, super tightly regulated rhythm.
So what is it exactly? Well, the cell cycle is just this orderly sequence of events a cell goes through to grow and then divide. And the goal is simple, but also pretty profound, to create two brand new genetically identical daughter cells from one parent cell. So to really get how it all works, we can break this cycle down into these distinct phases. You can almost think of them like a cell's 4 seasons. Each one has a very specific job to do before the cell gets the green light to move on to the next. Let's walk through them. First up is the G1 phase, or gap 1. This is where the cell just grows bigger and stockpiles all the energy and materials it's going to need. Next is the supercritical S phase, which stands for synthesis. This is where the cell carefully duplicates its entire DNA library. After that, it hits the G2 phase, which is kind of like a final quality control check, proofreading the new DNA. And then, the grand finale, the M phase, or mitosis, where the cell physically splits into two.
But, not every cell is on this constant loop of division. Some of our most important cells, like our mature neurons or hard muscle cells, they actually exit the cycle. They pull off onto a resting ramp called G0. They're still working hard doing their jobs, they're just not prepping to divide anymore.
And this brings us to the most fascinating part. This whole thing is not an unstoppable loop. It's an incredibly controlled process. So, how does a cell actually know when it's time to go from G1 to S, or from G2 into that final division?
The answer lies right here, cell cycle checkpoints. Think of them as incredibly strict security gates. There are 3 main ones. At each of these points, the cell literally pauses and asks itself a bunch of questions. Is all my DNA intact? Am I big enough to divide? Are my chromosomes lined up perfectly? If the answer to any of these is no, the whole process slams to a halt.
So who are the molecular guards at these checkpoints, giving the thumbs up or thumbs down? Well, there are 2 types of proteins, cyclins and cyclin-dependent kinases, or CDKs for short. These are the master regulators of the whole show.
And their partnership is really cool. Think of it like this. The CDKs are the engines, but they're turned off by default. The cyclins are the keys that turn those engines on. And here's the clever part. The levels of different cyclins rise and fall throughout the cycle, so a specific key is only available at a specific time to turn on a specific engine. For example, you need cyclin D to turn on CDK4 to get through G1, but you need a totally different key, cyclin B, to turn on CDK1 and kick off mitosis.
Now, to make this all even clearer, there's a really powerful analogy that everyone uses. Think of this whole control system like you're driving a car. You need an accelerator to go, but just as importantly, you need brakes to stop.
And this isn't just a convenient metaphor I'm making up. It's exactly how scientists think about these molecular controls. The things that say stop, those are the brakes in the system.
So here's the bottom line. The genes that make the go signals, the positive regulators, are called Proto-oncogenes. That's your accelerator. And the genes that make the stop signals, the negative regulators, are called tumor suppressor genes. Famous proteins like P53 and RB, those are your brakes, designed to stop everything if something goes wrong. So, in a healthy cell, you have this perfect balance of acceleration and braking. But what happens when that control system breaks down? What happens when the accelerator gets stuck down, or the brakes just fail?
Well, that is the molecular story of cancer. A mutation can turn a proto-oncogene into an oncogene. Suddenly, the accelerator is jammed to the floor. Or a mutation can shut down a tumor suppressor gene, and now your brakes are gone. Either way, the result is the same. The cell just divides and divides, ignoring all the stop signs. And that cellular pileup is what we call a tumor.
And if you want to know just how vital those brakes are, get this. That tumor suppressor, p53, its gene is found to be broken, mutated, and useless in more than half of all human cancers. That's staggering. It's like finding out that over 50% of all car crashes happen because the brake pedal was just gone.
So with this incredible understanding of the cell's own control system, how do we even begin to fight back against this runaway process? Well, the key is to be able to see what the cycle is doing in real time.
And one of the most powerful tools for this is a technique called flow cytometry. What it does is pretty ingenious. It uses a special fluorescent dye that sticks to DNA. So the more DNA a cell has, the brighter it glows.
And what you get is a graph that looks just like this. Cells in the G1 phase have a normal amount of DNA, so they create that first big peak on the left. Cells in G2 or M have doubled their DNA, so they're twice as bright and make that second, smaller peak. And all the cells in between are in the S phase. This graph is a literal snapshot of a cell population, letting scientists see exactly what's going on. And that really brings us to the final, crucial thought here. The cell cycle is this incredibly delicate balance between go signals and stop signals. Cancer is what happens when that balance is shattered. So the ultimate challenge for medicine is this. How do we design drugs that can fix the brakes or ease up on the accelerator of only the runaway cancer cells without shutting down the healthy cells that need this fundamental rhythm to keep us alive? That is the question that drives so much of cancer research today.